There we go. Welcome everybody to the CCEH sponsored Microscope Monday. We're excited to have Ken Dunn present to us today. Before Ken speaks, I want to take a moment and share my screen to talk about the Centers of Excellence in Hematology. Give me just a minute to do that. Okay, this is the website for the Centers of Excellence in Hematology. If you go here, there are several things that might be of interest to you. The first one that I think that is always of a big interest is what are the centers? They offer many services, and if you click here, there's core services across the country that can help you with your research. And if you just go to this site, you can kind of surf through the different cores that are available. For example, if you have a question about how to model what you see in your cells in animals, you can click on this link and click on live animal modeling, and you'll find that Yale and Cincinnati and the Fred Hutch in Indiana all have core facilities that can assist you in animal modeling to confirm what you're seeing in cells. The second place I want to guide you to is this funding link right here. So if you click to funding and just scroll down a little bit, what you'll find is the CCEH offers two types of funding opportunities, Type A and Type B grants. These are pilot and feasibilities. The Type A essentially is core credits at any core you're interested in using that you can bank up to $12,000 worth of credits. The Type Bs are a very typical P&F project, and we have a call out currently that's due August 1st. So I highly recommend you visit this site to learn more about it. And if you think that you have a good project or have a question, you're welcome to reach out to me and I can answer that for you. I'm going to stop sharing right now. And then I want to just introduce Ken Dunn, who comes to us from Indiana's CCEH. Ken got his bachelor's degree at UC Santa Cruz, and if you hopped on early, you might have heard us talking about the ocean there. He then went on to his Ph.D. at Stony Brook in New York, and finally did a postdoctoral fellowship at Columbia in the lab of, if any of you are endocytosis experts, in the lab of Fred Maxfield. And he has since been on the track of being an expert in microscopy and the development of novel tools to enhance your research in using microscopy. And with that, I'm going to turn the time over to Ken and let him share his screen. This is an interactive seminar series, so please feel free to stop and ask questions if you have them. If you're more comfortable populating them in the chat, I'm happy to monitor that and invite you to ask your question at that time. So thanks everybody for your attendance. And these are a great educational seminar series. So I hope you'll share this opportunity with others to come and listen. Okay, Ken.
All right, good. Thanks, Diane. Yeah, I want to second what Diane said, that I would really love to hear questions from people. I hate nothing more than giving an hour-long or 55-minute-long talk in a room by myself to nobody. So it'd be great to hear from you occasionally over the course of this thing. There's going to be some things going to be confusing to you, so I expect people to be open about that and talk to me about it. So my presentation's title was Intravital Microscopy, but I was realizing I'm going to be emphasizing multiphoton microscopy. And the other thing is there's lots of kinds of intravital microscopy. I'm going to be talking primarily about rodents. So today I'm real excited to give this talk. I love this. I love the topic and I really enjoy it. So that's the first problem. Great. So anyway, it turns out intravital microscopy has been around for a while. Marcello Marpighi was one of the first to look at blood flow in arteries and veins. He couldn't see capillaries. Anthony van Leeuwenhoek actually saw capillaries, saw capillary blood flow. And so since there have been microscopes, there's been intravital microscopy. So why would you want to do intravital microscopy? It's challenging. It's hard to do intravital microscopy. So one of the most important reasons for doing intravital microscopy is that it captures the in vivo relationship between cells and structures. And you're not going to get that in cultured cells. It provides the complete context of an animal with all the physiological inputs. And probably, I think, the most exciting thing is that it gets very complicated. The milieu, if you will, of cell biology gets really complicated in the context of pathology, or things like drug-induced liver injury. You can't really reproduce all of that. And so the idea of doing cell biology in the context of all those in vivo inputs is, to me, the most exciting aspect of intravital microscopy. This is a quotation I found. Most pervasive fallacy of philosophic thinking goes back to the neglect of context. And those of us, I mean, I spent my career studying polarized MDCK cells, which were pulled out of a Cocker Spaniel in 1959. And then I tried to extrapolate onto human disease. And so I'm as guilty as anybody. Why would you want to do intravital versus fixed tissues? And this is an important thing, because a lot of people come to me wanting to do studies they could just as easily do in fixed tissues. Well, one of the obvious things is you want to look at cell dynamics, things like immune cell migration, interactions between cells, cell and probe localization. The cells in the bone marrow are not really fixed down very well. And so once you fix the tissue, you're not entirely sure they're really where they were in vivo. A lot of small molecule probes. We look at siRNA sometimes, and they don't stay put. So if you actually want to find out where these probes are, you have to look in the live animal. Tissue function is going to be something I'm going to be spending a lot of time on. Things like microvascular flow. That's the preponderance of what people used intravital microscopy for, was looking at microvascular flow, permeability, organ function. And I'll be talking about kidney transport and liver transport. This is something that I've done in collaboration with various companies over the years. So the first thing I want to bring up is the idea of inferring versus observing the functioning of the kidney. And Bowman made these beautiful drawings of the kidney in the 19th century. And it was based on some drawings that Malpighi had put together. And from that, they derived a model of how they imagined that filtration would work. And I started giving these talks. We bought a 2-photon microscope around 2000. It was one of the first commercial systems available. And we collected these images of the kidney, and I'll start the movie in a second. And when I first showed these to movies full of nephrologists, it was like it caused an audible gasp in the room. Because basically what you're seeing here is the process that you imagined, but you never actually saw. I mean, everybody understood, everybody knew that it had to work this way. You have these small molecules that end up going in through the capillaries. They're filtered out through the glomerulus into the Bowman space, and they go down into tubules. But this was the first time people had actually seen it happening. And there's a lot of things. This is an example of something that we knew to be true. We were 99.99% confident of it. But there's a lot of other processes you can't see, that you can't evaluate, you can't imagine unless you actually see it in person. So the first studies that people started doing with intravital microscopy were kind of derived from the people who developed 2-photon microscopy, and I'll get to that in a second. But there's been a lot of studies of the brain. And so there's things like intravital calcium imaging. And this is in the brain of behaving mice. And I'll explain that term in a minute. And then another set of studies that primarily from Brad Hyman and Brian Baxkai, looking at the dynamics of amyloid deposition in the brain, which turns out to be hugely dynamic. It's not a progressive thing completely. It's not a monotonic increase in amyloid deposition. Another thing that people use a lot, and John Condilis really led the way of this at the Einstein, looking at cancer development and in metastasis, migration of cancer cells. And Peter Friedel and Grigorovich, I'm mangling that name, both actually came out of John Condilis' lab. And this is an example. I mean, Peter Friedel, this movie up here, if you can see my mouse, the migration of the cells is actually done as a group. Condilis came up with this way of using a photo convertible probe to identify cells at a particular time and then look at their migration over time. These are cells that actually end up dispersing into the lung from a mammary fat pad, and this is brain growth and vascularization over time. So the emphasis here is on longitudinal studies, in vivo studies of dynamics, progression of injury, progression of disease. And then the other aspect, the other focus, if you will, of a lot of intravital studies has been in the field of immunology, where people look at things like in vivo cell dynamics. And this is an example of interaction between different cell types, even sharing the material between different cell types. And then this is an example of a movie of migration of neutrophils towards a site of sterile injury. And so again, this is the kind of thing you can't analyze any other way other than intravital microscopy. we've been doing intravital microscopy, like I said, since around 2000, when we got one of the first 2-photon systems, and we've been using it for studies of kidney disease. And we've been funded since 2002 as these Indiana O'Brien centers. And one of the first observations we made is that albumin, in fact, is filtered. It doesn't make sense, given the size of albumin and the abundance of albumin in the blood. You would like to imagine that the designer wouldn't have it being filtered by the kidney. In fact, it is. And it turned out that surprisingly, the basis of the diabetic nephropathy in at least one model was not a defect in glomerular permeability, but in fact, it was a failure of uptake in the proximal tubule. So this right here these are endosomes that have internalized albumin. So this is an image, a cross-section of an image from a kidney. That's a glomerulus, it's a distal tubule, these are proximal tubules, and this is the S1, the initial segment of the S1 segment. You can see the albumin has been filtered and internalized, something that was real hot topic in the field for a long time, pissed off a lot of people. But the point was that they found out that not only was this kind of an interesting thing, but actually this was the basis of some of the component of diabetic nephropathy. Pierre Dager has advanced using intravital microscopy to characterize sepsis-induced kidney injury. And if I go through all of these things, I'm not going to have time to go through my talk. So it just suffice to say that he discovered that LPS is filtered by the kidney during sepsis and internalized by S1 segments, which signal to S2 that induces a reactive oxygen that's detectable, and turns out by FLIM, fluorescence lifetime microscopy. Since that funding, that we also get funded as a diabetes center. And we have a microscopy for the diabetes center, and working with them, we've been using various biosensors in vivo to look at things like the signaling of phospholipase D in insulin resistance. This is muscle. This is second harmonic of and this is a PAS biosensor of lipase D activity. This is glucagon-mediated PKA activation using a FRET biosensor in the liver. And this right here is Amelia Lindemann has done a lot of work with reactive oxygen, and she's looking at the role of reactive oxygen in beta cell death. So working with Enrique Serrazani at Vanderbilt, who's interested in the alterations of signaling that occur in the context of diabetes and how they may lead to enhanced susceptibility to infection in diabetes. So these are cells that are expressing the PKA biosensor, again, AK4.1. And Chris Ricehouse and various other people are interested in calcium dysregulation in diabetes. So this is the calcium transients following a glucose bolus. I've been doing a lot of work on intravital microscopy of the liver, and this image, as much as anything, is just to give you a sense of the resolution that you can get in intravital microscopy. Each one of these green spots here is a mitochondria, and is labeled with rhodamine 1, 2, 3. These studies were conducted, however, in collaboration with Lilly Research Labs to look at the distribution and the uptake of a therapeutic antibody, and you can epithelial cells in the sinusoids. Working in collaboration with the group in Bloomington, we were looking at acetaminophen hepatotoxicity. And this is another example of why you want to do intravital microscopy. Right here is a field of cells, that's bad. This is the liver of a living rat that's been injected with rhodamine 1, 2, 3, which fluoresces and accumulates in active mitochondria, fluorescing bright green. And also propitium iodide, which labels the dead cells because it becomes membrane permeant. And so all your dead cells are red, and these areas of darkness are places where the mitochondrial membrane potential has been disrupted. So what you see is these 2 hours after treatment with acetaminophen, you have all these dead cells that's surrounded by these regions of defective polarization of the mitochondrial membrane. What we didn't expect to see, however, is that within 2 hours, all these regions have recovered. And so a lot of the things interesting about acetaminophen hepatotoxicity is what is behind the fact that in some cases you recover, in some cases you don't. So this is a pretty fundamental observation. And again, it took intravital microscopy, seeing the vitality of the individual cells to detect this transient effect on the cells. People today, I assume, are here primarily because they're interested in the bone marrow. And Nadia Carlesso, who has since moved on to the City of Hope, developed a core facility to do intravital microscopy of the bone marrow niche. You shave the skull, you make a incision in the scalp, and you can image the calvarium of the mouse bone, bone marrow space in the calvarium. And this is an example of an image collected from, this is a bunch of images after injection of a rhodamine dextran and expressing GFP lyse cells and myeloid precursor. Individual panel from this thing looks like, and this is what the image looks like in a live animal. And Nadia developed a technique. I mean, this is not a new technique by any means, but she exploited the technique of using bone marrow transfer. You take a marked donor, and that could be, in this case, expressing GFP lyse, but it could also be any number of mutations or transgenics that could be derived from. You extract the bone marrow, you transplant it into an irradiated recipient, and you watch over time how the bone marrow space repopulates. And this technique, which is described in this paper down here, I really recommend everybody go have a look at if you're interested, has then been used in various studies in collaboration with Ruben Kapoor to look at the how loss of RAP1 creates a vacant niche that promotes repopulation, how Notch enhances, the loss of Notch enhances myeloproliferation in the bone space, when that was from Nadia and again, Ruben Kapoor. And then Eddie Swore and Katetti did a study looking at that, demonstrating that CD166 is critical to HSC graft. So again, this is all a matter of these transplant studies done in various transgenic mice models. The other thing that Goshen Kamoshka in the core was responsible for developing is the ability to evaluate how various treatments like age and radiation affect the function, the microvascular barrier functions. You essentially quantify the ratio of two dextrans, a small one or a large one, either in the vasculature or in this interstitial space, quantify that over time. And they, again, were able to use that technique to demonstrate the role of neuropeptide Y. Why else would you want to do this? Well, when you compare this to other in vivo, there's a lot of in vivo imaging techniques, and many of them are much simpler to do than intravital microscopy, but nothing that gives you the kind of cellular and subcellular spatial resolution, so the second and sub-second temporal resolution, nor the capability for multiplexing. So these are, again, I'm coming from a nephrology background, so I apologize that so many of my emphases are on kidney, but this is the kind of image you get from bioluminescence. You can basically see there's two kidneys there. MRI, you get a lot more detail, but you're not getting down to cellular resolution at all. PET, again, you can distinguish basically the cortex from the medulla, but in this kind of image, you can resolve individual endosomes, in this case, lysosomes. Lysosomes were labeled with a cascade blue dextran, and endosomes were labeled with I think this is actually gentamicin, but I'm not 100% sure, but you can distinguish these individual compartments. So the resolution is for all practical purposes, really equivalent to what you can get in cultured cells. So how do you do intravital microscopy? You're now talking about doing biology outside of a dish. And believe me, my background's in cultured cells, and it was great working with cultured cells, and it's not great working with animals. It's way more complicated. You now have live rats and mice in a dark microscope room. The first thing you have to think about is how you maintain animals under physiological conditions. In fact, the first thing you have to deal with is IACUC. You're going to have to write some good IACUC protocols and explain what you want to do. You have to consider anesthesia for both survival and non-survival studies. You use different forms of anesthesia for these things. And again, I want to point out, well, I'll in a second, some references for you to look at for more details about this. Probably one of the most challenging things has to do with the surgical preparation. For survival studies, it has to be aseptic. But I think the thing that limits most people's ability to do intravital microscopy is the kinds of microsurgical techniques you have to apply in order to make it work at all.
Hey, Ken.
Yeah.
This is Diane Ward. Just a couple of questions about this first piece. Do you guys share, or is there a site that shares, sort of the wording for IACUC and these protocols?
Yeah, we could do that, actually. We've done that, but it's never been formal across the nation. So that's a really good idea. Because it's true. I mean, if you come upon the term, the way that IACUC committees like to see it, we would like to just reproduce that rather than having it coming out from whole.
Every investigator would be interested in accessing something like that. Yeah.
Yeah. Good. No, that's a great idea. I hope I remember it.
I will help ask again. Thanks.
That was good. Terrific. Thanks. Yeah, the other thing, once you've got the animal on the microscope stage, you have to maintain the physiological state, temperature, fluids, eye drops, keeping the preparation moist, keeping the animal hydrated. You have to monitor the animal physiology, his respiration, heart rate, temperature. And one of the things I want to point out that's kind of subtle is that when you think about temperature, people think about warming this microscope stage, but the microscope objective, if you ever touched one, they're cold and they'll take the heat right out of your finger. And so to the degree you've got the whole animal warmed up, but you've got this objective, immersion objective right up against the tissue, what you're doing essentially is putting an ice cube on the tissue where the place you're imaging. So you have to think about those things. This is another quote from Marcelo Marpighi. For the unloosing of these knots, that is, elucidating these problems, I have destroyed almost a whole race of frogs, which does not happen in the savage, whatever that word is, of Homer. That, by the way, was a parody written not by Homer. It has to do with a war between frogs. These are the two papers I want to refer you to. And this is recorded, so you can come back and see these things later. So Nadia Carlesso and her group published a Joe video describing how to do all these different things. George Rose wrote an article recently about how to do intravital microscopy and the surgical preparation of rats and mice. So the first issue is, how do you access the tissue of your interest? The first studies of intravital microscopy were actually done, the first 2 photon studies of intravital microscopy that I saw were done by labs that were not really physiology labs. And I found a surprising number of intravital microscopy of human skin, which I think might have been postdocs. I'm not entirely sure of that, but the point is that if to the degree you're looking at something other than the skin, you're probably going to have to do something to get at the tissue you're interested in. So the dorsal skin fold model is one most people are familiar with, and it's transparent. It's very easy to work with if you're looking at the vasculature. If you want to look at the brain, there's 2 approaches, and there's a tempest in a teapot about which is the right approach. You can either put in a cranial window, or alternatively, you can just thin the bone down to a thickness equivalent to a cover slip. And people have different thoughts about which of these is the best way to do it. You can put a window in, and this is an abdominal imaging window we'll be talking about later. Or alternatively, you can just simply expose the organ for microscopy, just do direct surgical exposure. This is how we get access to the calvarium. Essentially, the mouse is it the video of it? You present it, you flip the mouse over, he's on a cover slip dish, and then this is a stereotactic device to immobilize the skull, and we can provide people with the design of this thing as well. And again, you keep the animal moist, you keep the animal warm, and then not only do you keep the animal warm, but you also have an objective heater. Another interesting thing that I am astonished at, I found a YouTube video that I since have not been able to find, but this is a very similar example. People make miniaturized 2-photon microscopes. There's an amazing example that came out of Stanford where they were using just stuff from cell phones and glass beads to make miniature disposable intravital microscopes. Anyway, the idea is you mount this on the skull of a mouse, use a micro endoscope to insert into the brain. And at this point, the animal is able to run around. And since this guy's expressing GCAM, I think 3, I'm not sure about that, you're looking at calcium transients at the end of that probe. And there's all kinds of experiments you can do by depending on where you put this in the animal. But again, this is an animal that's not under anesthesia. This is an animal that you can give them various sensory inputs. You can show them things. And the video that I found on YouTube that was so cool is they had this video run, this animal running on a trackball, and they were showing him a movie of what he thought he was running through, and they're recording all his neural activity at the same time. So this is another way of getting optical access to your biology. The other things that you have to think about is that if you're going to be doing micron and submicron resolution imaging, you can't have your biology moving around. It has to be immobilized to submicron levels, which is not easy. And the other thing that you have to think about, another challenge of it, is the optical nightmare of living tissue. Last week, Jörg Berensdorf talked about how all the things you do to ensure that you're collecting images without any kind of aberrations. And there's things you can do as long as you're looking at something that's sitting right on a cover glass. To the degree you're imaging into biological tissue, you don't have a vote. You don't get to fool around with it. You have to take what it gives you. And we'll talk some more about both of these things later, the optical nightmare of living tissue. What kind of microscope can you use? Well, it turns out you can use any of a variety of microscopes, and they all have their strengths. And so it really comes down to what do you got in your center. This is an example of intravital wide field microscopy. There's that dorsal skin fold. You can have transillumination. There's a transillumination image, or alternatively, you can do epifluorescence. And people do use this a lot for study of endothelial microvascular function. It has some real serious strengths. Wide field microscopy can collect images on the order of 100 frames per second. And I hope this movie plays. Good. This is an image collected at a volume collected from the liver of a living rat, collected at 100 frames per second. And this is 1 minute. And the thing that was interesting about this movie is that you're collecting the images fast enough. We developed some software for basically essentially continuously monitoring blood flow in all these different vessels. And when we did that, we found there was this amazing variability. There are blood vessels. This is red blood cell velocity. There's some vessels that stop entirely and then start again. There's some vessels that start in reverse direction. But it's when you read about measuring microvascular flow, people make measurements and say that's how fast the blood was flowing. Well, that's how fast the blood was flowing in that capillary at that point in time. It's very, very difficult to integrate over time and space to really get an overall number, if you will. So the complexity of microvascular flow is captured by this wide field microscopy approach.
It kind of looked like the flow was due to something that was blocking some of those vesicles or those vessels, and then it cleared. Is that not?
Yeah, it could be. Yeah, I mean, there's white cells, for example, and sometimes you would actually see a shadow where it's blocked, and you see it start up again. Before the advent of two-photon microscopy, people used intravital confocal microscopy, and there's advantages and disadvantages to that. One of the advantages of it is it's a lot less expensive, and it also allows you to do reflection microscopy. Chip Montrose, who's now in Cincinnati, I believe he's in Cincinnati, use intravital confocal microscopy to look at the intestine in ischemia reperfusion and injury in the intestine. Another form of confocal microscopy is something called spinning disk confocal microscopy. Spinning disk is very cool. Rather than having a single laser point that you scan over the sample, you have I forget how many it is. I think it's like 10,000 points that you're scanning over a sample and simultaneously collecting 10,000 points of image. This means you can collect images at like 100 frames per second. You also illuminate the sample at very low levels. And so Cubes, Micah, what is Cubes? Great, I don't have his name here. Wonderful. I forget Cubes, Paul Cubes, there you go. Paul Cubes has been the person who's primarily used this technique. This is an example of how he used this in the kidney to look at macrophages in interacting with Staphylococcus aureus. And the next video is an example of something we collected using two-photon microscopy. And you don't compare this to speed. In fact, spinning disk can collect images much more rapidly. But what I want to call your attention to is the enhanced resolution we're getting out of this versus spinning disk, and also the ability to do multiple colors. One of the problems with the spinning disk system is that you collect each image sequentially, which slows the whole process down. So let's talk about multiphoton fluorescence. This is where you're going to have to indulge me because I really love talking about this, probably more than people like hearing about it, but what are you going to do? This was something that when you think about a Jablonski diagram, you can have, this is typically how you see fluorescence occur. You have an absorption, which takes an electron up to a higher energy state. It loses a little bit to vibration, and the remaining energy is released as a photon as it goes back down to ground state. Maria Gompert-Meyer in 1931, who subsequently got the Nobel Prize, postulated that fluorescence could be stimulated by the simultaneous absorption of multiple low energy photons. So for example, if you have two photons with half the energy of this guy over here, if they arrive simultaneously, you can excite an electron at a higher energy state and you get the fluorescence back out again. It turns out that when jumping ahead of myself a little bit here, but here's the two photon fluorescence excitation spectrum of a bunch of organic probes, as well as a bunch of GFP labeled molecules. And what you can see is that the energy is roughly half, twice the wavelength that you'd ordinarily use. And when you do that, they all overlap with one another such that a single excitation wavelength can excite multiple fluors. This is an example of Hertz labeled nuclei in a live animal, a live rat. Hertz labeled nuclei. This is 500 kD fluorescein dextran. No, excuse me, 10K rhodamine dextran, which has been filtered. It's in endosomes and distal tubules, and a large molecular weight green dextran, which is in the vasculature. All this is collected with a single excitation at something on the order of 800 nanometers. So the trick here is that there's this simultaneous absorption of two photons. This was postulated in 1930. The problem is that it takes an enormous density of photons in order to make that happen. In other words, having simultaneous absorption means they have to be absorbed within 10 attoseconds. You have to arrive within that volume within 10 attoseconds, which is 10 to the minus 17 seconds. So detectable two photon fluorescence excitation takes something on the order of 200 gigawatts per centimeter squared, which is 300,000 times the photon flux at the surface of the sun. So it wasn't until 1961 that someone actually built a laser that was capable of doing that kind of thing and demonstrate, yes, you can in fact stimulate fluorescence that way. This is something that one of the leaders of two photon microscopy put together years ago when it was first described, Winfried Tanks, one of the inventors of two photon microscopy. And the idea is, well, that's great. How often does this actually happen in nature? And this is a guy dumping, I don't know, probably rhodamine something into a pond. And he made some calculations that if you had a molecule of rhodamine be exposed to direct sunlight, it'll absorb a one photon absorption about once a second. A two photon absorption will occur once every 10,000 years. And a three photon absorption has never occurred in the history of the universe. I'm going to show you examples of where two photon and three photon fluorescence has been used in a microscope. How do we do that? Well, the first thing you do is you increase the photon density by focusing it down through a laser to a high numerical aperture. So now you've got it constrained to a very small femtoliter volume. And this was first demonstrated in 1978. So the only place you have this really, really high density of photons is right at the focus. Now this is very cool because what this means, since two photon excitation decreases with a fourth power of distance from that focus, it means you're not stimulating fluorescence anywhere else except at that focus. And this is an example of a cuvette of a fluorescent probe that's being illuminated from one side with visible illumination and from the other side with IR illumination, which you can see is the fluorescent. Oh, I'm showing myself, but I'm not showing you. I'm sorry. You can see the fluorescence is only coming from this one point in the cuvette by two photon excitation, whereas you have the standard hourglass shape of excitation coming from visible laser. So how do you make an image with two photon microscopy? Well, you collect a 2D image by raster scanning, just like you do with confocal microscopy. You make a 3D image by making a stack of these things. But the difference between confocal and two photon microscopy is whereas with confocal microscopy, you have to have a filter over here to eliminate all this out of focus fluorescence. With two photon microscopy, all the fluorescence is only coming from that one point. You know that. You don't need to put a pinhole in anymore. So if that means the fluorescence is spatially constrained, we don't even image the emissions anymore. You excite the fluorescence from one point and you map the emissions you collect back to digitally to where you want it to come from. So there have been various ways of collecting light. In other words, you don't have to image the fluorescence. You just have to collect it. So Warren Ziffel came up with this really ornate way. I don't even know where the sample is here. Where did the sample go? The objective, the specimen's there. And you've got all these detectors. You're surrounding the thing with detectors. You can do this with parabolic reflectors onto two different detectors. And then finally, Enrico Gattone came up with a way you just put a PMT on the far side of the sample and you collect all the light. You're just collecting it. And as long as you're scanning a point, you know where to map it in the sample. what this means is that if you don't need to filter it, it means if you don't need a pinhole to reject the out-of-plane fluorescence, you now can collect all the light. You're not susceptible to scattering anymore. This means that this is really, really good for doing deep tissue imaging. With a confocal microscope, the deeper you go, all your light gets scattered, none of it makes it to the pinhole, and then all of a sudden you lose all your contrast. With 2-photon, you just simply have to collect all the light out of that tissue as best you can. Now, earlier, I was telling you that we have to illuminate the sample with 300,000 times the power of the surface of the sun, which if you've done microscopy, you know it's not probably a really bright idea. So the solution, which was derived in like 1990, came from pulsed lasers. And the idea is that rather than having this continuous laser at very, very high power, you have a laser that you're pulsing at 80 megahertz, very, very briefly, 100 femtosecond pulses. And this is Watt Webb's fathers, the people who actually invented multi-photon, 2-photon microscopy by putting all this together. Now, what that means is that you have peak power sufficient to stimulate fluorescence, but the duty cycle is so low that the average power is very, very low. 80 megahertz, there's 100 femtoseconds, pulses 80 microseconds apart. This means that the duty cycle is equivalent to 1 second pulse per day. In other words, it's illuminated only 0.01% of the time. When you're looking at the laser, it's mostly off. And the effect of that is that although you're putting this enormous power in, it's so brief that if you had a 100x NA and 1.4 objective, the volume is so small that continuous illumination would warm the water only 0.2 degrees C. So it accomplishes the high peak excitation you need, but not the heating. So a couple of cool things about the serendipity of laser design. At 80 megahertz, the laser pulses are spaced 3 fluorescence lifetimes apart, which is very cool because it gives all the fluorophores an opportunity to relax back to ground state, get more photons out. But there's also encounters, a lot of people like to explain 2-photon excitation in terms of fluorescence lifetime. It has nothing to do with fluorescence lifetime, completely different orders of magnitude. And the other thing that's cool is that, and again, during the course of a 100 femtosecond pulse, a fluorophore will be illuminated 320 times during a 4 microsecond pixel dwell time. This gives you 320 times to collect fluorescence without any kind of saturation problems. So you get multiple excitation relaxation cycles. And this all comes out from the fact that the original lasers happen to be a certain size. And it was serendipitous that that size corresponded to this 80 megahertz pulse rate, which happens to be just pretty much ideal for fluorescence imaging. I'm not going to talk about this because I've got too many things to talk about. 3-photon excitation is another way that you can do fluorescence excitation. So now it requires 3 photons arriving simultaneously, which is even harder. This requires very long wavelengths because now you're essentially having 1/3 the energy that you would have for single photon excitation. It allows you to do deeper imaging. This is an image all the way down to 1.2 millimeters. The problem with it is that it requires 10-fold higher power than 2-photon. To get a laser to do that, you have to have, and to not blow up your sample, you have to have a very, very low repetition rate. So now all of a sudden you're at a slow enough repetition rate that in order to collect the photons, you cannot collect the frames very long. So while Chris Zhu has demonstrated how valuable this technique is for imaging, it's something that has some serious limits as well. Why would you want to do intravital? Why would you want to use multi-photon microscopy? Well, one of the advantages of multi-photon microscopy, aside from the depth, and I'll get to that in a second, is that it allows you to do other nonlinear modalities. So this is an image volume on the left that I collected from mouse liver. And this right here, the first thing you saw was second harmonic image from collagen. This is something that is, it's a non-centrosymmetric structure such as collagen will provide the sense of contrast. There's no photo bleaching involved with it. And you can complement 2 different colors of fluorescence with that. This is an example of a volume that we collected. This is not intravital microscopy because it's human. This was a human sweat gland that was pulled out of the skin of a human donor. And all this white material up here is collagen, second harmonic. All this red here is actually from a mitochondrial membrane potential sensitive dye. So this allows you to do not only fluorescence, but also second harmonic. And Peter Friedel's kind of led the way looking at third harmonic, which is another nonlinear mode of imaging that's based on refractive index differences. So you can see things like interfaces between lipids and non. So there's an example of 2 different colors of fluorescence, as well as second harmonic and third harmonic images collected from a live animal. Why else would you want to use multiphoton microscopy? You can image UV dyes without UV illumination. I showed you this a little while ago. This is Herx labeled nuclei and Cascade Blue labeled lysosomes. An advantage that a lot of people point out is that the photobleaching is less, and it kind of is, but it's kind of not. If you think back to that volume I showed you before, this is a hunk of fluorescent plastic. I think Dave Piston put this together, that he's illuminated with a laser beam used on a confocal microscope. And what you can see is that over time, this region, there's the sample that he's focusing on, but you get photobleaching above and below that. On the other hand, when you do 2 photon excitation, you only have photobleaching of that one focal plane. So for collecting 3D volumes, this is a very, very cool technique because you're not getting this accumulated photobleaching effect. That said, there's some very strange things that happen with photobleaching at 2 photon microscopy that people don't understand. And in fact, the photobleaching at one focal plane is frequently worse than by 2 photon than by a confocal. What are the limits to photon microscopy? Deep tissue imaging is easier because you don't have scattered light. Scattered photons can be imaged at something on the order of confocal resolution. This allows you to do intravital microscopy. So in a lot of ways, it's really the ideal approach for intravital microscopy. So now I'm going to quickly go through the challenges and limits and solutions to intravital multiphoton microscopy. Again, it's an optical nightmare. You can't match the refractive index because you don't have one refractive index. You got all kinds of different refractive indexes in your solution, and so in your tissue. So there is no matching it. You can match the average refractive index, but you can't do it in the way that you'd like to. The tissue is a scattering mess. I mean, particularly the kidney. These tubules act like lenses. They're objects in that act like mirrors. And then the other thing is that for things like doing adaptive optics, all of this stuff's moving around continuously. So there's no good solution to it. Motion artifacts from respiration and heartbeat. These are probably the thing that most people stop doing intravital microscopy immediately because of this. You have to, as I was telling you, you have to mobilize your sample as best you can. And there's various techniques that we've described for doing that. This is a movie collected from the liver, and you can see we've got terrific stability. All these nuclei here from the hepatocytes are completely stable over time. You can see a lot of immune cells moving around. This is an example of preparation we did in a mouse, where we had, again, enormous stability. Each one of these endosomes is absolutely stable. This is very unusual, though. More often than not, we have problems with motion. This is a particularly egregious example of motion. On the left is the original volume, and then on the next 2 panels are after being put through a processing technique that we developed for doing image registration. One is just linear registration of the sample to get rid of this migration of the field. And the other one is this non-rigid registration, which eliminates the distortions that are happening within the frame. And it's been a very powerful technique that allowed us to do quantitative analysis of things that otherwise would not be possible. Limited speed. And a standard galvanometer-based scanner, we can collect images on the order of 1 to 4 frames per second. You can go faster with a galvanometer, but not a lot faster. There are other architectures. So you can use a polygonal mirror, you can use a resonance scanner. Many people have access to resonance scanning confocal microscopes. What I want to point out, though, is that once you go up to a 30-fold higher frame rate, you're going to have a 30-fold shorter pixel dwell time, which then means you need to have 30-fold higher excitation in order to keep the same signal to noise. You have to have an enormous amount of fluorescence in order to put up with that. Your dwell time is so short, you're not collecting very many photons. So these are a single frame image of dextran flowing through a capillary, and you can see the shadows of individual cells. You can see endosomes, all kinds of things. This is collected at 1 frame per second. This image down below was collected at 28 frames per second. And you can see the signal and noise is awful because there's just simply not enough signal. I want to point out that when we load up dextran into the vasculature, that's a very, very strong fluorescent signal. So I would say that It's unlikely that in a lot of cases you're going to be able to actually get 30 frames per second from a live animal and have it be meaningful. And the reasons are that the signal is not there, but even if it was, your power is limiting. The multi-photon excitation is frequently done at close to saturation. So even if you increase the laser power, you don't get more signal out. And then the other thing, as I was mentioning before, is that photobleaching increases faster than signal. Another approach to doing high-speed multi-photon microscopy involves parallel scanners like the spinning disc I was telling you about. And there's some really interesting architectures for doing that, and they work really, really well. Instead of having a single point scanning over the sample, you have a bunch of points scanning over the sample, and you image onto a CCD. Problem with that is that you are now dividing your excitation up into a bunch of fluorescent points. You may not have the power to divide it up among different illuminations. And then the other thing is that if you image the scattered emissions rather than mapping them onto their points of excitation, if you think about it, the reason that two-photon microscopy works for deep tissue imaging is that we're not imaging. What we're doing is we're collecting the photons and we're exciting them at a particular point. If you excite it everywhere and collect the image, you end up getting this mush. So yes, you can have a multi-photon microscope, but you can't use a multi-point multi-photon microscope, but you can't use that to image deep into tissues because you are now susceptible to the effects of scattering, just like you are with a confocal microscope. Limited signal and signal attenuation with depth. This is something that people don't really fully understand. And a lot of people that talk about two-photon microscopy just kind of overlook this as an issue. This is an image volume collected through a kidney. What you can see is it's very bright at the top of this glomerulus. But as you get back down to the bottom of the glomerulus, it gets dimmer and dimmer and dimmer. In fact, we can probably image something on the order of 100 microns into the kidney. This is a problem because glomeruli in most rats and mice are more than 150 microns. So what this means is that for most animal models, we can barely get to the functional unit of the kidney. And this is even more of a problem in things like the brain, where things are just way too far away. So what are the limits? What limits the reach of multi-photon microscopy? This is some image. This is collected by Sentence and White a long time ago, 1998. This is one of the first confocal two-photon microscopes. And this is the kind of loss of contrast you have with depth with a confocal microscope. The resolution you get, loss of contrast with depth with two-photon microscopy. And so you can see that you're getting an image down to 40 microns here, whereas you're really not getting much of an image at all, even at 20 microns with confocal microscopy. But it's not a deal breaker. You know, the point is that this is not enormously much more depth the way it's typically presented. And the reason for this has to do with the fact that you're still limited by light absorption, you're still limited by light scattering to some degree, and you're limited by spherical aberration. I don't have time to go through this, but let me just say, if you just run some numbers you find in the literature with respect to how much light is absorbed by biological tissues, the excitation emission, it turns out that, you know, the excitation is reduced in millimeters. It's just not a big deal. Light absorption is not an issue, except for things like hemoglobin and melanin, which can make a huge difference. Light scattering, on the other hand, is a much bigger deal. Excitation is attenuated by 75%, anything between 70, as little as 70 microns, and emissions can be scattered by 50% in anywhere from 14 to 70 microns. So even though these emissions are still largely collected, light scatter is the thing that's limiting the depth of imaging. How can you address that? Well, you can reduce the optical aberrations. And last week, Jorg mentioned how spherical aberration works. The different points of light are focused at different points of the sample. This has the effect that if you image using an oil immersion objective, you image in an aqueous medium at the surface, you have a great point spread function. You go 100 microns into the solution, you can't image it anymore. The fluorescence is there if you stretch it, but you can't see it. Whereas if you match the refractive index, you can image deeply into the tissues. So matching the refractive index is an important thing. So here's an example of how we use that in intravital microscopy using a water immersion objective. This is endosomes, 22 microns, 61 microns, using a silicone oil immersion objective that matches the refractive index better. Now we can get basically equivalent images at either 20 or 67 microns. Alternatively, you can use adaptive optics, which is the more innate approach, and we don't have time to talk about that today. You can use longer wavelengths of light. And so that you have less scattering, less absorption, a lot of things at longer wavelengths of light. So you can extend the depth of imaging. And that includes things like 3-photon microscopy. You turn the laser up, and this is a really, really interesting thing. People imagine you could just keep turning the laser up, and what happens ultimately, and again, Winfried Denk's lab demonstrated this. When it ultimately happens, the whole idea of two-photon microscopy is that you only want to stimulate fluorescence from this one point. And as long as the photon, and the fact that the photon density is so low here because of the size of the radius of the beam, you don't excite any fluorescence here. However, if you start having to turn the laser up in order to get enough photons into depth, you actually start to generate fluorescence at the surface of your organ. And the thing that ultimately limits the depth to which you can image by two-photon microscopy is the point at which the background fluorescence equals the fluorescence that you're getting from the stimulation. And that's all has to do with the fact that this fluorescence from up here in the volume is increasing with laser power such that you can no longer see the things. And so there is a hard limit to how deeply you can image by two-photon microscopy. It's dictated by the distribution of fluorescence in your sample. There are things you can do. There are horrible examples. You can cut into the tissue. You can stick a gradient index lens into the tissue. Now, I understand that if you do that, you're not going to be able to browse. You're going to look at wherever that thing's poking into the tissue. And then finally, Mike Levine came up with this really, really elegant approach using prisms that you can insert into the tissue. And then you bounce the light off the side of the prism to image axially. And so this is a conventional XY image, it's XZ image, beautiful resolution. The problem with these things, these prisms are incredibly tiny and really difficult to work with. So we didn't get very far with it. Another challenge of subcellular imaging living animals is longitudinal studies. This is an example of an abdominal imaging window that was developed by Jaco van Rienen's lab. It came from Condilis's lab. And this is a 3D image volume collected from the kidney of a living mouse. And the first thing you see in this volume is a second harmonic from the capsule of the kidney. And then below that are the various GFP light cells that are attacking the cover slip, if you will. And then below that is some dextran that's been injected into the vasculature. I want to point out that these white cells that are attacking the cover slip, they can affect the biology. So there's something to remember that when you're using these windows is that you're causing an inflammatory response. And then the last thing I want to talk about in the remaining few minutes is how do you extract quantitative data from intravital microscopy, knowing that the amount of fluorescence you detect is a function of depth. You never really know where you are in the volume, to be honest. It's challenging to keep track. Not only that, but you also don't know how the tissue is actually affecting signal as a function of depth. So how can you get quantitative data? Well, you can quantify without intensity. You can look at structural changes over time. You can quantify motion, as I showed you a moment ago, where again, you're not quantifying the amount of fluorescence so much as using it as a mechanism of contrast. This is an example of quantifying neutrophil migration. And again, you don't necessarily need intensity information yet. You can also then quantify incidence. You count things that are above a certain threshold of fluorescence. And again, this is all done by Paul Cubes, looking at white cell attack of lasers injured region. You can use ratiometric measurements. These are measurements of protein kinase A that I think I showed you earlier. This is after adding glucagon, you measure the ratio of YFP to CFP fluorescence. This is independent of focal depth, so you get constant values regardless of depth. Ratiometric measurements, again, this is what I showed you earlier in terms of measuring the fluorescence in the vasculature versus the free space. So look at vascular permeability. And another way of thinking about it is in time series measurements. So this is an example of the effect of a drug on transport in the liver. There's fluorescein that's been injected into the vasculature, and over time it's transported into the hepatocyte, and then finally the bile canaliculi. Since you're quantifying everything relative to this time zero time point, you end up getting constant values over time that are independent of depth. Last slides, I just want to say that there's some places you can get more information about intravital microscopy, the bone marrow niche. Nadja wrote a really nice article in 2017 that it's this Jove article in video. More recently, there's been a couple of other papers referred to here. In particular, Christine Lacelso has done some beautiful work of intravital microscopy of the bone marrow space. And those of you interested in intravital in multiphoton microscopy, you can refer to several websites as well as some really cool papers. Most of them at this point relatively old. And with that, I'm happy to take questions.
Thank you, Ken. I think I want to ask the question maybe for the audience, which is, if you have an interest in doing intravital microscopy, but maybe not the capability at your center or your institute, how can people figure out what they can do about that? Like, does the Indiana Corps do microscopy for investigators?
Yeah, yeah, and it being a new core, we're still working out the details of that. But the point is, the funds that you were talking about that are available to CCEH investigators are available to support these kinds of studies. So we have the people, okay? We have the resources. We've got 3 2-photon microscopes. We've got people that can do the surgeries, people that do the microscopy of the calvarium and all these other organs as well. It's just a question of coming up with the funds to compensate them, because the center doesn't actually have the funds to fund all the studies. And so by and large, the way this would work is that there would probably be some pilot studies that we would conduct with someone to find out if there's any there. And at that point, it would be a matter of applying for funds to fund the studies. The first thing, though, is that people contact me and see, does this have legs? Is there a reason that we would want to do intravital microscopy? Is it feasible? Is this something we might want to think about doing? And then once it passes that test, then the next step is to look at doing some pilot studies. And then after that, to try to come up with funding. The idea with the CCEH funding is that would be something that you would use to get preliminary data for an honest to God R01 proposal that would use this kind of thing. And the other thing I want to point out is that many of you are at institutions that have 2-photon microscopes. They may or may not be used for intravital microscopy, and that might be a barrier. But in my experience, there's a lot of people that bought 2-photon microscopes aren't doing much of anything with them, and we would love to see people doing something productive with them. So the other thing that we can do is we can help investigators work with their local facilities to see if they can implement 2-photon intravital microscopy at their institution.
Cool.
I know.
Is that Jonathan?
Yeah. Hi, Ken. That was a beautiful talk, and
Thanks.
the work is so beautiful. I think we discovered a iron transporter, and when we knock it out, we lose endocytosis, which is a little bit confusing as to why that would be. But the problem in the kidney is that the S1, S2, S3 are completely different, and so on. So how would one actually do intravital microscopy and actually mark the segments at the same time and know exactly where you are?
So Alan Hall, I could Pierre David did this too, but Alan Hall, I think he's published this. Anyway, it's actually really embarrassingly simple. You do a bolus injection of something that's filtered, and you watch where it goes. And the first thing it goes to is S1, and then S2, and ultimately S3. Now, assuming they're all represented in the focal plane you happen to be camping out on, you can distinguish them. And then once you've done that sequentially, you've also discovered there's differences in autofluorescence, these characteristic differences in autofluorescence you can then go forward with. So we can distinguish those pretty well. And again, I would suggest you look at Alan Hall, Andrew Hall, I always call him Alan Hall, Andrew Hall in Europe. He's got some papers describing how this is done and how to do it. So you can do that. Now, the issue then is the question of whether or not all those are going to be present at the depth that you want to look at. So for example, S1 is really easy to find in a Munich blister rat that has superficial glomeruli, because there it is attached to the glomerulus. It's more difficult to distinguish these things in mice, but like I said, it can be done.
And do you take collaborators for purposes of grant writing, et cetera? And
well, John, you're a nephrologist, so you could apply for money from the O'Brien Center. So that's the other thing we have here. So there are 3 different. So we do intravital microscopy with the CCEH, but we also do it for the Diabetes Center and the O'Brien Center. And the O'Brien Center in particular is to serve national researchers, to provide this as a service nationally. And so the funding mechanism in the O'Brien is in the air right now, to be honest. But that would be probably the best approach, and that everything's in place for that currently. So that for kidney microscopy, which I guess what your project doesn't sound like a CCEH project, if I'm not mistaken, but it would be.
No, except that it is an iron metabolism, which it's
good.
right.
Great, good. We get double credit. Yeah, no, we should talk about that, because that sounds completely feasible to me.
I look forward to reaching out to you. I'll send you an email. Thanks so much.
Great, sure.
Other questions? Anybody's welcome to chime in. I populated in the chat where you can find these recordings, and Dr. Dunn's recording will be up in the next day or two, so that you can find that, or you can watch the first one that was presented by Dr. Biersdorf.
And I also, I don't think I shared my email address, so I don't know. That's the other thing. I really, it's difficult for people to evaluate this from a talk like this. So if you're interested, I really encourage you to email me. I don't know. Is there some way that we can?
I'm just putting it in the chat right now.
Perfect. Good. Great. Because again, I'm very excited about working with people to implement intravital microscopy for their research. It's something, like I said, I've been doing it since 2000 for the renal research community, for the Diabetes Center for probably the last 10 years. And I'm looking forward to doing it for the CCEH.
Great. Anyone else?
Right. All right. Well, thank you for attending today and listening to my talk. Look
Yeah.
forward to hearing from you.
Thanks, Ken.
Thank you.
Okay.